For decades, biology has been studied in bulk. It's like analyzing a city by blending all its buildings and people into one. You get average data but lose all the nuance. Single-cell analysis changes that. It lets us study individual cells one by one, unlocking detailed insights into how tissues truly function, how diseases evolve, and why therapies succeed or fail. What impact do you think is going to have on patients in the next 10 years?
We see this potential of using single-cell methods, single-cell multi-omics methods to really improve therapeutic options, either as a companion diagnosis eventually for more advanced therapies, or to promote different novel therapeutic methods to be developed. So, but if we think even a little bit further, I think this kind of very fine resolution we could get from single-cell biology, this would also enable us to detect maybe small changes in healthy people that could eventually lead to disease a couple years down the way. And this would also enable us to have better health management and also to prevent even some diseases from happening.
Europe, US, and in China, how far are we in terms of seeing these capabilities integrated into everyday clinical decision making?
When we founded the company, we were already very much aiming for the eventual application in a more clinical setting. Yeah.
In terms of application, there are more promising breakthrough in neurology.
Like the brain is so amazing. Using single-cell technology to develop better therapy is autoimmune disease. This is really like getting to be a hot topic since autoimmune diseases also come at different forms. And also longevity, our new Abu Dhabi joint venture is also focused on. And this
founded in 2018, 2017, then you raised about 150 million US dollars, several large VCs. But I understand that to create a company of this caliber, it takes more than just great science and great passion for science. What do you think have been the key drivers for growth?
All right. Quick message for me. If you're interested in the life science tools and equipment sector and you don't know where to start, it feels overwhelming, or you simply want to compare notes, check the Bio Innovation series I created for you so that you and your team can develop your own strategy to get the most of this exciting field. Enjoy the rest of the video.
Welcome, everyone. I'm here with Dr. Nan Fang, CEO of Singleron. Thank you, Nan, for joining.
Yeah, thanks, Fabio, for this very interesting and exciting opportunity to chat.
Thank you thank you also for your kind of hard work. It's 4 p.m., like 20 past 4 here in the UK. So I guess it's 11:20 there in China. Oh, yeah, 22 in China. So thank you very much for finding the time. So now, as I anticipated, so before we dive into the science and technology of your company, of Singleron, I'd love to hear more about your personal journey and what originally sparked your interest into single-cell genomics and how that led you to become the founder and CEO of Singleron.
Mm-hmm. Okay, so I have always been a biologist since I chose biology to be the subject of my study for the bachelor, because I really like the complexity of the biological systems. And I also really like in my university in China, University of Science and Technology of China, the biology students were required to study physics with the physics students from the physics department, chemistry with the chemistry students, and math with the math students. It really liked the whole experience of really diving deeper into different subjects of science and use that to study biology, because biology is just simply so complex. And after that, I got my PhD in immunology at University of Iowa. I worked with Dr. Gary Kresge there for T-cell biology. And as we now, if we look back, we start to realize actually the immunologists have always been single-cell biologists, because if you think about all the immunology studies and methods, we use FACS to look at cells at single-cell level. We look at like several protein markers from each single cell. We use those markers to separate them. And we do immune histology, and we stain cells, and we look at them also under microscope at single cell level. So it's probably also a little bit natural, of course, for an immunologist to someday become single cell biologist. But before that happened, I also had 10 years experience at QIAGEN in Germany. So I worked on product development. I started with developing PCR products, and then also qPCR products, RT products for both life science and IVD applications in 2011, 2012. And QIAGEN decided to go into the NGS market. I was also tasked with setting up the product development for universal NGS products, like the sample and library prep products for NGS. So that was also a great experience, enabling me to get deeper into the genomics field, getting to know also, this quite new field at that time. And we also worked already on single cell sequencing, but just at that time we launched actually the first single cell sequencing related products, the Repligy single cell library prep kits in the market. But at that time it was really like single cell was that you had to pick one single cell after another, either use FACS sorter, use your own hands. And that was and then you probably you could analyze maybe 10 or up to 100 such single cells at one time. It was already giving people, I would say, a lot of new information that you couldn't say at bulk level, but still, if you only could analyze 10 cells, you really don't get enough statistic power, especially after you see all the cells are so different from each other. So then in 2015, there were several publications coming from the US, first describing the high throughput single cell technology, where you could really first barcode each individual cell, and then you analyze like thousands of cells together, and using those molecular barcodes, you can separate the information to single cell level, but still you get thousands of cells at once. And that's really the time point where I started to think the single cell technology or single cell sequencing at this kind of high throughput fashion could enable us to better understand biology, and also especially understand biology in health and disease, and to enable us for more precision medicine than the precision medicine we know, like by looking at mutations and so on. So that's also why in end of 2017, together with my co-founder, Dr. Nan Fang from Yale University, we just really had a couple, not so many, maybe 2 or 3 very deep discussions. And we just decided, okay, looks like this is a field we would really like to get into. We do have the expertise and technology needed. For example, this microwire-based technology Ron developed in his lab at Yale, and without much thoughts, we just decided to fund the company. So the company was then funded in 2018. And since then, I have been quite dedicated single cell biologist, I would say. Yeah.
Well,
So that was the journey.
Well, it's a great story, and it's certainly led by your passion for, as you said, a biologist ends up being a single cell biologist, and then let's say fast forward 7 to 8 years now, Singleron raised about 150 millions. Like with large investors like Arch Ventures, SoftBank, Vision, China Venture, and Lilly Asia Ventures, and CDH Innovation.
All right. Quick message for me. If you're interested in the life science tools and equipment sector and you don't know where to start, it feels overwhelming, or you simply want to compare notes, check the Bio Innovation series I created for you so that you and your team can develop your own strategy to get the most of this exciting field. Enjoy the rest of the video.
So as you mentioned, single cell genomics essentially is changed the way we study biology, because we're not looking at the bulk, but we are looking at the single cell. From your perspective as founder and CEO of Singleron, what excites you the most about where this field is heading, and what impact do you think is going to have on patients in the next 10 years?
Yeah, I always believe the measurement technology or detection technology and therapeutic technologies, they really co-evolve. For example, if we now have a higher resolution measurement analysis technology like single cell approaches, we would be able to see ourselves or the patients, the people in a much higher resolution. This would enable us to develop also better therapeutic methods. So that's one thing, but vice versa, if you have an advanced therapeutic strategy, such as cell therapy, such as immune therapies, those are like more living therapy. It's a more systematic therapy. It's different from maybe a target therapy that's already quite good. That's targeting one or several mutations. Here you really target the whole human as a system, and to predict which human being could respond to this kind of systematic therapeutics and which humans might not. I think you also need a different type of analysis to give you the biomarkers, so that for you to predict, for example, the treatment outcome. So that's how I see the highest potential, and also what really excites not only me, but also all of our colleagues at Singleron, because we see this potential of using single cell methods, single cell multi-omics methods to really improve therapeutic options, either as a companion diagnosis eventually for more advanced therapies, or to promote different novel therapeutic methods to be developed. So that's only one part. Of course, that's related to people who are already like patients who would need treatment. But if we think even a little bit further, I think this kind of very fine resolution we could get from single cell biology, this would also enable us to detect maybe small changes in healthy people that could eventually lead to disease a couple years down the way. And this would also enable us to have better health management, and also to prevent even some diseases from happening, because we could have interventions before like certain health situation would lead to disease. So that's, I think, a longer probably vision and also a longer possibility, but I do believe that would also become reality if we further develop this kind of high resolution biology methods.
Yeah. Some therapeutics might be more effective if we catch the disease earlier on. They might be able to work and be efficacious only if we administer in time. So if you consider like Europe, US, and in China. How far are we in terms of seeing these capabilities integrated into everyday clinical decision making?
Yeah, I think I have to say, of course, because if we have to deal with patients, we need to be especially careful. That's why the clinical field is normally relatively conservative regarding the new technology. And it always takes time for new technology to move from basic research to translational research, and then eventually to the real clinical applications such as a diagnostic tool. So we are still not there yet. I have to say there are still things needed, such as accumulation of more evidence and more data and more verification of this technology before it could become a routine, for example, diagnostic test. But I see a lot of efforts already from the clinical field, from also, of course, the research field and industry like us moving to this direction. One thing is, for example, in EU, in Germany, we are part of the Certainty EU Horizon 2020 project. And the Certainty project is first to use single-cell multi-omics data from multiple myeloma patients to build this so-called digital twin of multiple myeloma patients, so that after this, with the data plus AI, you build this digital twin, and then you can use this model to really predict for the future blood cancer patients the possible treatment response, if they take drug A or they take drug B. So this is really, the project was started 2 years ago, and we are moving along quite as planned. And we are on track in already discussions and planning of commercial applications of all the data we have collected in the last couple years with the consortium. So I think there is a clear motivation and also clear track of moving to more clinical settings. So that's one example. And also in China, for example, I think people are probably also a little bit less conservative, I would say, regarding some molecular diagnostic new tools. So we are also on track to get a Class 2 medical device certificate for our instrument later this year. And we are already discussing, sometimes approached by quite well-known hospitals developing LDTs for certain patients with single-cell technology. Yeah, so although there are not yet certified FDA approved assays yet, but I think there are already a lot of activities, and it's, I would say, like NGS field maybe 15 years ago, like people are really hugging this new technology, and they would really like to move it to the clinical settings since they see the potential.
Yeah, now we can be more optimistic, let's say now. So
yeah.
Great. So Singleron is one of the fastest growing player in this field, in the single cell genomics. Can you help me understand the differentiation between Singleron and, let's say, how Singleron is positioned in the single cell genomic space, and how you're thinking about shaping this field's future?
Yeah. Because when we founded the company, we were already very much aiming for the eventual application in a more clinical setting with our technologies. We actually, for our product design or development, we have always taken into consideration the cost effectiveness, that it should be relatively easily automated, and also scalability into consideration. So that's how we, for example, chose microwire-based chips as the base of our single-cell and labeling technology. And because it could really, from the cost point of view, it could really one day be reduced especially with scale and also with in combination with the magnetic beads we use, it can be relatively easily automated from the beginning to the end. So those, like this kind of focus in potential clinical application, especially routine clinical application, and focus on the cost as well as standardization, I think is probably one thing that we probably do differently from other single cell companies. And another one I would say is that we probably also work more on single cell multi-omics, because we see that if you have a single cell that you would like to analyze, there's not only RNA, DNA, there's probably also protein, there's metabolites, there's probably also other analytes in the cell that you would like to analyze as much as possible with one assay. So that's also another direction we have been also focusing on, that I think we also have some quite unique products and advantage.
Excellent. So yeah, it's more positioned towards a clinical application, and in order to make it happen, you need to design the product accordingly. And I think I read that everything that you design is tailored to work with any kind of setup, from the very manual to the fully automated.
Yeah.
And now in terms of applications, I think in terms of application, in general, single cell genomics, we started with cancer, then immunology, then certainly now there are more promising breakthrough in neurology, where the cellular diversity and spatial context is very important. And how is Singleron approaching this field, this kind of application in particular, and what role can your tools play in unlocking like the brain complexity, essentially?
Yeah, I agree. They are like, the brain is so amazing. And I think if I hadn't chosen to do my PhD in immunology, neuroscience would be like my second choice, because it's also as complex, almost as complex as immune system, and also almost as exciting as immune system. And but anyway, and it's really also, as you said, it's complex, then it needs probably also this kind of high resolution biology technology like single cell. Yeah, and we do have products that are tailored for neuroscientists to study their objects better. For example, we have developed certain tissue dissociation solution that could also dissociate brain tissues quite well, since normally brain tissues are not so easy to be dissociated to single cell suspensions, because they tend to aggregate together. They have a lot of debris, and if you don't get a good single cell suspension, of course you cannot really analyze them well. So we do have some products that we develop to be also tailored for a neural system, but we are also involved actually in another European project that's called B2B Rare. It's a project for the rare neuronal disease, where like this is a consortium including University of Düsseldorf, as well as a couple other universities in the Western German area, and also Singleron. And where we do is also use single cell multi-omics technology to analyze the biopsy from rare neuronal disease patients that, based on the current knowledge, don't really have a standard therapy. And with the data coming from different omics data, the doctors are really hoping they can repurpose certain existing drug to treat those patients. Yeah, we are providing also our single cell technology there, and we are also very excited to see the potential clinical use. Actually, the first patient as proof of principle already got his treatment with the data from multi-omics analysis, and he really got much better with this repurposed drug. That's also one big reason how we got the grant from the state of NLV in Germany to continue with this kind of approach of using single-cell multi-omics and other omics to do a drug repurposing for rare disease, neuronal disease patients.
And that's great, because I've seen certainly an increase in interest for even developing drugs or repurposing drugs in neurology and in psychiatric disorders. It wasn't the case back, I think, in 2018, 2019, but certainly now there is much more interest. We had a few successful exits, and that's probably that significantly increased the appetite, and there are certainly lots of unmet needs there. And hopefully this technology is gonna work towards that, towards enabling many more solutions for these patients. Beyond, so oncology, immunology, neurology, three big areas. Beyond these applications, have you seen any other less obvious area where single cell might still have an impact maybe in the next few years?
I think another area we also see like growing interest of using single cell technology to do sample analysis and trying to develop better therapy is autoimmune disease. This is really like getting to be a hot topic since autoimmune diseases also come at different forms and formats. And also it involves like certain tissue types plus immune cells very often. So that's also where we see the power of single cell biology, single cell analysis in giving people more insights for them to either identify or discover new drug targets, new therapeutics. So that's another area. And also longevity, I think it's also a very interesting area. Actually, what our new Abu Dhabi joint venture is also focused on. And but longevity actually is a very broad concept. It can include immunology, it can include probably also chronic and a lot of different things. But here, I think if you, for example, one project we are currently planning is really to screen the blood cells of healthy people and then to do some like follow up at different time points with if they undergo certain lifestyle change, and then we would also like to monitor whether their immune cells would change with this kind of different lifestyle interventions. And this is actually quite exciting also application. And other than that, basically because I would say single cell analysis is a very basic and fundamental biological tool now that can analyze both can be used in both cell biology and molecular biology. So anything in biology that you want to analyze or want to study, theoretically, you can use this tool.
It's like a base that can be used if you understand the cell, what is happening there at cellular level, and the communication we can understand much more. And you basically covering all the essential area, all the latest frontier. Longevity itself covers several diseases. Now let's switch gear. I know it's very late, Ijaya. So the very last question for me would be that at the beginning you described your personal journey to becoming the founder of CEO of Singleron, and then let's say I summarize everything by saying yes, and then founded in 2018, 2017, then you raised about 150 million US dollars, several large VCs. But I understand that to create a company of this caliber, it takes more than just great science and great passion for science. So I was wondering maybe what do you think have been the key drivers for growth, and how have you managed to prioritize them along the journey from 2018 till today?
I would say our team would be the most important factor in moving us so fast. Because I think we have very probably common understanding inside of the company that what we are doing is really aiming to improve health. And we also see from our daily work by in all those projects, in all those conversations with our customers that we are really indeed also doing what we are aiming to do. And that's one thing. And also another thing I would say a very large percentage of our colleagues, when they joined Singleron, this was their first industry job. But I think that's probably, of course, on the one side, they need to learn a lot of things. But on the other side, I really do think this is an advantage because then they have no fears. Probably if you have, sometimes I worked in other company, I probably could think, here maybe we cannot do this because we tried this before and this is probably not working. But then my colleagues would just tell me, well, but maybe that's your old experience. For us, they don't have this kind of baggage from the history. They would just like to try everything. They don't have any fear. For them, nothing is impossible, regardless whether it's technology, it's developing a new approach, and whether approaching certain customer that I would even think, no, they are not going to be interested. They are not our target customer. No, this is probably not going to work. Yeah, but maybe we can just try. Sometimes they don't even tell me, they just try. And then they went back with the results. And I was like, oh, I didn't know that would even work. So I think this kind of really fearless naivety of our team is really,
perspective.
Yeah, and also the learning attitude. I think although they probably didn't really already have the industry experience, but everyone is also eager to learn. And I think that's together with their courage, their fearlessness. And I think, I would think that is the largest factor probably for us moving relatively quickly until now.
Well, and I remember from the quick exercise of the Mr. CEO that you rarely take leave, but then for maternity, that was the longest leave you ever took, for 3 years, which means, and the reason why I bring it up is because it means that you combine being a mother and your family with your successful career. So it's again a sign that it's possible to combine both. There might be times where you had to prioritize your family over your career, but then you came back and even stronger than before. So
yeah. Thanks. Yeah, I believe so. So you just have to, you have different priorities at different stages of your life. That's also what I tell our colleagues. That's also when they also need to take maternity leave. I also just let them know, okay, if you would like to focus on your family, on your kid at this time, it's really perfectly fine with me. And because I was like that before. And but what I would at least expect from myself and also from other people is, but if they decide to do certain things, whether it's being a full-time mom or whether it's work, just for the time when they are doing that, maybe the devotion would be also quite important.
Well, thank you so much